Bismillahirrahmanirrahim. This is Dr.Naeem Khan, Chemistry Department, Cohort University, and I'm instructor for the course Carbohydrate Metabolism with code CHEM548, and this is first online lecture. Today we are talking about pyruvate metabolism. In today's lecture, we will discuss about the possible fates of pyruvate metabolism. What are the possible routes in which pyruvate can undergo onward breakdown? Then out of these, we will go to anaerobic metabolism of pyruvate because there are some aerobic pathways and some anaerobic. So we will go to anaerobic metabolism and will highlight some of the problems associated with the anaerobic metabolism. Then we will discuss out of the anaerobic metabolism, we will elaborate in detail the conversion of pyruvate to lactic acid, which is one of the anaerobic metabolism pathway. We will calculate the total ATPs that will be produced in the anaerobic metabolism of pyruvate to lactic acid, and in this way we will try to calculate the total number of ATPs that will be produced from one glucose molecule. Lactate dehydrogenase enzyme is very important enzyme in this pathway, and we will have a thorough look of this enzyme. Then we will come to the lactic acid cycle. We will discuss how this lactic acid synthesis in the body is cycled in the from muscles to liver, and later on how it is consumed. And finally, of today's lecture, we will elaborate the importance of the conversion of pyruvate to lactate. Before starting the today's lecture, let us to have a revision of our last lecture. Last time we discussed glycolysis, and we know that in glycolysis, from 6 carbon compound glucose, we reach to 2-3 carbon compounds pyruvic acid. And in this process, from glucose to pyruvic acid, 2 ATP molecules were consumed, while 4 ATP molecules are produced. So it means there was 2 net ATP molecules produced. At the same time, 2 coenzyme NAD are reduced to NADH. So this means that up to now, starting from the glucose in the carbohydrate metabolism in glycolysis, we have 3 carbon compound pyruvic acid and 2 net ATP molecules produced, and also 2 reduced NAD coenzymes. Okay. Now to start with, the first point, what are the possible routes for the carbohydrate metabolism? So far, there are detailed literature available about the carbohydrate metabolism, and 4 of them are the most abundant ones. And these are, first one is the conversion of pyruvate to lactate or lactic acid. This is anaerobic metabolism, which means this process is not requiring oxygen. Next one is the conversion of pyruvic acid to alcohol, which is also known as alcoholic fermentation. And this is also anaerobic mechanism. Third route, well-known route, is the conversion of pyruvic acid to alanine. Alanine is an amino acid, and here the conversion of pyruvic acid to alanine is providing a window of the synthesizing proteins from pyruvic acid. Remember, pyruvic acid is not a carbohydrate, but it is the product obtained from the glucose metabolism in glycolysis. So it means here we are having a route of synthesizing proteins from carbohydrate. And third, and the main pathway is the entry of pyruvic acid into tricarboxylic acid cycle, which is also known as Krebs cycle. And this is through pyruvate dehydrogenase pathway. Actually, this is the main mechanism which is taking place in the living things, and this is the oxygen requiring pathway. I mean, this is aerobic pathway. Under the aerobic condition, the pyruvate pass by this pathway. What are the problems with the anaerobic metabolism? In the last slide, we saw that there are two pathways, anaerobic pathways of the conversion of pyruvic acid into lactic acid or alcohol. So these are not the main pathways or the recommended pathways. And as I mentioned that the main pathway is the passing of pyruvate to Krebs cycle, the last one, which was, and that was aerobic. Because the anaerobic pathway has a problem. What problem? Look, in glycolysis, the reduced NAD coenzymes are produced from oxidized one. Under normal condition, this reduced NADH are oxidized in the electron transport chain, and oxygen is consumed, and in this way hydrogen passes from the NADH and water is synthesized. But under the anaerobic condition, if oxygen is not available, it means that NADH cannot be oxidized to NAD. So it means in the body there is a calculated amount of NAD available. So if all NAD are converted to NADH in glycolysis, and if they are not being converted to NAD, so it would mean NAD are consumed, no more NAD. So if no more NAD, because all are converted to reduce, this would mean glycolysis will stop. And remember, in glycolysis, we are having 2 ATP molecules produced, though 2 ATP is not a big amount, because from 1 mole of glucose, normally under aerobic condition, we are having 36 to 38 ATPs, but at least this is a part of energy. So the anaerobic metabolism, the serious problem is glycolysis produces the NADH, which cannot be oxidized, so it is stored. And when all NAD oxidized form are consumed, even the glycolysis will stop. So the small percentage of energy which was releasing will now be stopped. In the anaerobic metabolism of pyruvate, one pathway was the conversion of pyruvate into lactate, and this is by this mechanism path. Here we can see that pyruvate is actually the oxidized form of the lactic acid. And in this conversion, which is catalyzed by the lactate dehydrogenase enzyme, pyruvate is actually reduced and the hydrogens are for reduction are supplied by the reduced NAD coenzyme, which itself undergoes and gets oxidized. The hydrogen are accepted, and by reduction, pyruvate is converted to lactic acid. And this is a reversible reaction, which would mean that the two substances, pyruvate and lactate, will be in equilibrium. And as per the Le Chatelet principle, any change in concentration of the pyruvate or lactate will shift the equilibrium in a direction so as to minimize the effect. So, if there is a change in condition, the pyruvate, the lactate can be back converted to pyruvate. How many ATPs will be produced? Remember, 2 ATPs will be produced in the anaerobic breakdown of 1 glucose. Why? These are the 2 ATPs which are actually produced in the glycolysis. No more ATPs can be produced, as in the last reaction here. We can see that no ATP is produced. So, and this is the end. So it means if we have anaerobic condition and the pyruvate produced in the glycolysis is converted to lactic acid, no more ATP will be produced. So it would mean that 1 mole glucose will give us 2 ATP molecules, and the two reduced NAD are big oxidized into NAD, and also instead of 2 pyruvate now we have 2 lactate. So it would mean that in the anaerobic metabolism of glucose, at the end we will have 2 lactic acid molecule and 2 ATP molecules. Yes, the reaction is fast and does not require any oxygen. Basically, it is anaerobic reaction. Lactate dehydrogenase enzyme is the main enzyme which was catalyzing that reaction of conversion of the pyruvic acid to lactic acid. Let's discuss some important points of this enzyme. This enzyme is actually an isozyme having different forms. For example, 5 forms are designated here as M4, M3H, M2H2, MH3, and H4. This M and H are actually the designations. Basically, this enzyme is a tetramer made up of 4 molecules, 4 polypeptide chains, which are coiled upon one another and making this protein molecule. And these 4 chains are actually of 2 types. M and H is designated because of their presence in muscles, skeletal muscles, and in heart muscles. So the 4 chains of 2 types, 2 M types or 2 H types, or depending upon the composition, 4 compositions are possible. M4, when all 4 chains are those which are predominantly present in the skeletal muscles. MH3H is also a type of enzyme which is present in the skeletal muscle. Similarly, H4 and MH3 are those forms of the enzyme which are predominantly present in the heart muscle. Remember, M and H polypeptide chains are different from one another on the basis of amino acid sequence. So they have 5 possible different forms, and these can be designated as such. This knowledge of the M4H3 has a very medical application. Look here. Skeletal muscles and liver contain predominantly the M forms. Heart contain the H forms. So look this. During and after myocardial infarction, I mean heart attack, heart cells die releasing LDH into the blood circulation. So it means in the heart circulation, the H form enzyme, H4 MH3 enzyme concentrations increases. So this is a medical diagnosis that if the concentration of this lactate dehydrogenase enzyme of the type H4 and MH3 concentration will increase in the blood circulation, it would mean that there is something wrong with the heart muscle. And vice versa. If there is a MA3H or M4 form increases, it would have clinical diagnosis for the some problem with the liver. So this concentration has important points. Next is the Cori cycle. Cori is a person who described this cycle for the first time. This is lactic acid cycle. The lactic acid produced from the glycolysis, from the glucose through glycolysis is an acid. So its deposition in the body is not recommended. In this cycle, whenever glucose is converted to glucose 6 phosphate, this is actually the first reaction of the glycolysis, and later on we will come to know that glucose 6 phosphate can be converted to glycogen as well, and in the form of glycogen it is stored in the muscle. However, under normal conditions, this glucose 6 phosphate is converted to pyruvate, which was our last lecture, and in today's lecture we converted glucose pyruvate to lactic acid. Now this lactic acid when passed to blood and when it reaches to liver, in the liver it is back converted to pyruvate and then converted to glucose 6 phosphate and then to glucose. Remember, in liver the main pathway is the conversion of glucose 6 phosphate to glycogen and it is stored. But whenever there is a low glucose level, this glycogen is broken down and it is converted to glucose, and glucose form it is passed back to muscle. In this way, this helps to maintain the glucose level normal in the body. For example, in fasting and the Sahri time and Iftari, when the glucose level is high, it is mainly stored as glycogen. But in the later hours of the day, when the glucose level decreases, this glycogen is broken down and glucose is formed. Liver Cori cycle, the liver releases some of the glucose. Yes, most of this is converted to glycogen, and glycogen can be broken down to glucose when needed, because this is the storage form, animal storage form of glucose. As starch is the plant storage form of glucose. What are the importance of this conversion of pyruvate to lactate? This help drive glycolysis. Yes, because in glycolysis, reduced NADH are produced, but in this pathway, the conversion of pyruvate to lactate, what it does, it is converting the NADH to NAD. It is oxidizing. So this NAD is then again available to glycolysis, and in this way, it helps the glycolysis not to stop. It is a reversible process and can be regenerated. Yes, the process was anaerobic, so any increase and decrease in one of the product, the reaction will go in the other direction. Look, if condition become favorable and oxygen become available, so pyruvate will pass to electron transport to the Krebs cycle and electron to electron transport. In that case, the pyruvate, the lactate will be converted to pyruvate. But in other case, if oxygen is not available and more pyruvate is synthesized, it will be converted to lactate. So this is a reversible reaction, and it has its importance to maintain the internal environment in a balance. Lactate fermentation is very important for those tissues which are having oxygen deficiency or oxygen normally their oxygen concentration is not so good, especially in red blood cell or retina or very special skeletal muscles. For example, in the a person is having a heavy exercise, there is a decrease in oxygen, so they are using this pathway for oxygen, for the energy production and loss. This is very important for the plants and microbes which are living in the absence of oxygen, so they are totally dependent upon this energy release. For today's lecture, Western Thought and Leninger book are recommended and elaborate all the points we discussed today in detail. And if you have any question, we may discuss. Thank you. See you next time. Until then, Allah Hafiz.